Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today we're going to cover Chapter 8 for our MCAT Biology playlist, and this chapter is titled The Immune System. Here we're going to cover the following objectives. First, we're going to look at the structure of the immune system. We're going to discuss the two main branches. The first is innate immunity, which is your body's rapid, non-specific defense. And the second is adaptive immunity, which is slower, but it's highly specific. We're also going to go over the key anatomical structures that make this system possible. Next, we're going to dive into the innate immune system further. We're going to start with the non-cellular, non-specific defenses your body uses to keep pathogens out in the first place. And then we're going to explore the different types of cells that are part of the innate immune response. After that, we're going to move on to the adaptive immune system. We're going to explore that further as well. Here, we're going to identify the major cell types involved. We're going to see how the adaptive system is activated. And then we're going to talk about how it distinguishes between self and non-self to protect against diseases while avoiding damage to the body's own tissues. Finally, we're going to discuss the lymphatic system. Here, we're going to cover its structure, how it connects to other systems in the body, and the important functions it serves. With this introduction in place, let's start with Objective 1 and look at the overall structure of the immune system. The immune system, again, has two main divisions: innate immunity and adaptive immunity. Innate immunity is also called non-specific immunity because it responds to a broad range of pathogens rather than targeting any one specifically. It's always active and it acts quickly to provide an immediate defense when pathogens are encountered. However, it does not generate long-term protection or memory against a pathogen. In contrast, adaptive immunity is also called specific immunity because it is tailored to recognize and respond to particular pathogens. This type of immunity is going to take longer to develop during the first exposure, but it has the ability to form memory cells, and that's going to allow for a much faster and stronger response if that same pathogen is encountered again in the future. Now, the immune system, it's not a single organ. Instead, it consists of multiple components that are located throughout the body. One major site is the bone marrow, where immune cells are produced. These immune cells are collectively called leukocytes or white blood cells. The thymus is another important site where T cells mature. The spleen is also going to play a critical role by filtering the blood, removing old or damaged red blood cells, and also just providing a site for initiating immune responses. B cells, which mature in the bone marrow, they're going to be part of the adaptive immune system. And when they're activated, B cells can then differentiate into plasma cells, which are specialized for producing antibodies. These antibodies help neutralize and eliminate pathogens. And this process is referred to as humoral immunity because the antibodies circulate in the body's fluids. T cells, which mature in the thymus, they're also part of adaptive immunity, but they function in cell-mediated immunity. So rather than producing antibodies, T cells are going to coordinate immune responses, and they can actually directly kill infected cells. Cytotoxic T cells, for example, they recognize infected cells and they destroy them to prevent the spread of infection. Another important structural component of the immune system is the network of lymphatic structures. Lymph nodes and lymphatic vessels, they circulate immune cells throughout the body and act as checkpoints where these cells can monitor for pathogens. In addition to lymph nodes, other immune tissues are distributed throughout the body. They include the gut-associated lymphoid tissue, or GALT, which contains Peyer's patches in the small intestine, as well as the tonsils, the adenoids, and the appendix. All of these structures, they help detect and respond to pathogens that are entering through various body surfaces. Now, leukocytes are produced in the bone marrow through a process called hematopoiesis, which begins with stem cells. These stem cells give rise to many different cell types, including those that make up the immune system. Leukocytes can be divided into two main groups, granulocytes and agranulocytes. Now, we've discussed this in previous chapters, but it is definitely worth repeating. Granulocytes are cells with visible granules in their cytoplasm, and they include neutrophils, eosinophils, and basophils. Neutrophils are the most abundant, and they specialize in fighting bacterial infections. Eosinophils are important for combating parasitic infections. They also play a role in allergic responses. Finally, basophils are involved in allergic reactions as well, where they can release histamine that contributes to inflammation. Agranulocytes, on the other hand, they lack visible granules and they include lymphocytes and monocytes. Lymphocytes are key players in adaptive immunity. B lymphocytes are going to be responsible for humoral immunity by producing antibodies, while T lymphocytes are responsible for cell-mediated immunity by coordinating responses and then destroying infected cells. Monocytes are going to be large phagocytic cells that circulate in the blood. When they migrate into tissues, they're going to differentiate into macrophages, which function to engulf and digest pathogens and debris, and also activate other immune cells. Macrophages take on specialized forms depending on their location in the body. So that is something that we're going to be able to get into as we transition from Objective 1 into Objective 2. So in Objective 2, we're going to take a closer look at the innate immune system. To start, this figure gives us a helpful reference on how to distinguish between innate and adaptive immunity. On the left, you're going to see the components of the innate or the non-specific immune system, which we're going to cover now in this objective. And on the right, you'll see components of the adaptive or specific immune system, which we're going to discuss in Objective 3. The innate immune system is our body's first line of defense, and it includes non-cellular, non-specific barriers that work to keep pathogens out before they can establish an infection. Let's begin with discussing the skin, also known as the integument. The skin, it serves as a physical barrier against bacteria, against viruses, against fungi and parasites. Its outermost layer, the epidermis, it forms a very tough shield that's difficult for pathogens to penetrate. In addition to this physical protection, the skin also produces antibacterial enzymes called defensins, which disrupt microbial membranes, as well as antimicrobial compounds that are found in the sweat. But any damage to the skin that happens, such as cuts or abrasions, those are going to compromise this barrier, and that can allow pathogens to enter the body. The respiratory tract, that also plays a critical defensive role. The lining of the respiratory passages is covered in mucous membranes that are going to trap particles and pathogens. And these membranes, they're equipped with cilia that beat in a coordinated fashion to move the trapped material towards the oropharynx, where it can be swallowed or expelled. The respiratory tract, it also contains lysozyme, which is an enzyme that breaks down the peptidoglycan cell walls of bacteria, which helps to eliminate them before they can even cause infections. Then the gastrointestinal tract, it uses both chemical and microbial defenses. So our stomach acid, it creates a highly acidic environment that kills most ingested pathogens. In addition, the gut harbors a large community of beneficial bacteria that compete with harmful microbes for both space and nutrients. And this competitive environment helps keep pathogens in check. However, if you're taking antibiotics to fight an infection, those antibiotics can disrupt this balance in the gut by also killing beneficial bacteria. And what this can allow is for the growth of antibiotic resistant organisms that can grow in an unchecked manner. But beyond these physical and chemical barriers, the innate immune system also includes the complement system. The complement system is a group of blood proteins that circulate in an inactive form, and it can be activated to then help destroy pathogens. Complement activation can occur through two main pathways. The classical pathway is triggered when antibodies bind to antigens on a pathogen surface, and that's going to link the adaptive and innate immune response. In contrast, the alternative pathway, that doesn't require antibodies, and it can be activated directly by pathogen surfaces. Now, regardless of the activation route, the complement proteins are going to work through a cascade of reactions that can opsonize pathogens, and that's going to make them easier for immune cells to engulf and then recruit additional immune cells to that site of infection and assemble the membrane attack complex. That's going to essentially puncture the pathogen's membrane and ultimately lead to its destruction. Another important component of nonspecific defense is the group of proteins known as interferons. These are secreted by cells that have been infected by viruses. And interferons serve as an early warning signal to neighboring cells. What essentially happens is they induce them to reduce protein synthesis and degrade viral RNA, which will hopefully limit viral replication. They also increase the presentation of antigens on the surface of infected cells, making them more visible to cytotoxic T cells. Finally, interferons help activate other immune cells, such as natural killer cells and macrophages, to target infected cells more effectively. So far, we've talked about the physical barriers of the innate immune system. But what happens when these barriers are breached and pathogens make it into the body's internal environment? At that point, the innate immune system turns to its cellular defenses, and this includes macrophages, natural killer cells, and granulocytes. Let's start with macrophages. Macrophages are large phagocytic cells whose main job is to engulf and destroy pathogens. They can originate from monocytes in the blood, which migrate into tissues and differentiate into macrophages. And they can be part of a resident population of macrophages that live in specific tissues. Now, macrophages are also antigen-presenting cells, or APCs. That means that after they engulf and break down a pathogen, they can display fragments of it called antigens on their surface. And they do this using special proteins called major histocompatibility complex, or MHC molecules. MHC molecules essentially act as a display case for antigens, and that allows other immune cells, particularly T cells, to inspect them and then decide how to respond. There are two main classes of MHC molecules, and the difference between them comes down to where the antigen originated from. MHC class 1 molecules present antigens that come from inside the cell. This is called the endogenous pathway. All nucleated cells in the body can use this system. If a cell is infected with a virus or it contains abnormal proteins, fragments of those proteins are going to be displayed on MHC class 1 molecules. And then cytotoxic CD8 plus T cells can recognize these abnormal displays and kill the infected cell. MHC class 2 molecules, on the other hand, they're used in the exogenous pathway. These molecules, they present antigens that originated outside the cell, such as bacteria or toxins that were engulfed. And this pathway, it's used by professional antigen-presenting cells, which includes macrophages, dendritic cells in the skin, some B cells, and certain activated epithelial cells. The antigens that are displayed on MHC class II molecules, they're going to be recognized by CD4 plus helper T cells, which then activate other parts of the immune system to respond to that threat. Dendritic cells in particular, they're considered the most potent antigen-presenting cells. They are found in tissues that are going to be in contact with the external environment and are highly efficient at capturing antigens and then migrating to lymph nodes to activate T cells. Finally, macrophages and dendritic cells, they have pattern recognition receptors, including a group called toll-like receptors. These receptors are going to detect common molecular patterns that are found on pathogens like bacterial cell wall components or viral RNA. And then what happens is they trigger the release of cytokines that help recruit and activate other immune cells. And this is going to allow the immune system to respond quickly, even before the adaptive immune system has had time to develop a very targeted response. Now, moving on to the next page, we've already discussed macrophages in detail, but to summarize, macrophages are large phagocytic cells that originate from monocytes. They migrate from the blood into tissues where they engulf and digest foreign pathogens, dead cells, and even debris. They also act as antigen-presenting cells where they can display antigens on their surface using MHC molecules to activate the adaptive immune system. In addition to destroying pathogens directly, macrophages release signaling molecules that stimulate and recruit other immune cells to the site of infection. Another important cell in the innate immune system is the mast cell. Mast cells are found in connective tissues and mucous membranes, and they're best known for releasing histamine and heparin, which cause blood vessels to dilate and become more permeable. This helps immune cells reach infected or injured areas a lot more easily. Mast cells are also involved in wound healing and defense against pathogens, but they're also central to allergic reactions. Next up, we have natural killer cells or NK cells. These are specialized lymphocytes that function in the innate immune system. They circulate in the blood and they can migrate into tissues. NK cells identify and kill cells that are infected with viruses or have become cancerous, especially when those cells have reduced or absent MHC class I expression. What they do then is induce apoptosis or programmed cell death in their targets without the need for prior antigen exposure. Dendritic cells are another type of antigen-presenting cell, and they are among the most effective at activating the adaptive immune system. They're going to be found in epithelial tissues such as the skin, lungs, and digestive tract. When they capture antigens, they're going to then migrate to lymph nodes where they present the antigen to T cells and initiate the adaptive immune response. Monocytes are going to be the circulating precursors to macrophages and dendritic cells. They are stored in the spleen and they move through blood vessels to reach infected tissues where they can then differentiate in response to inflammation. Neutrophils are the most abundant type of white blood cell, and they represent the first line of cellular defense in the innate immune system. They're going to be rapid responders that migrate quickly from the blood into infected tissues. Neutrophils kill or inhibit bacteria and fungi using toxic granules and enzymes, and they also recruit other immune cells to that site of infection. Basophils, although less common, they're going to play a role in defending against parasites and also in allergic responses. They circulate in the blood and they release histamine to promote inflammation. Finally, eosinophils are involved in combating multicellular parasites and also certain bacterial infections. What they do is they release toxic granules that can kill pathogens, but can also cause a little bit of collateral tissue damage. They're also associated with allergic diseases like asthma. Now, with that, we've been able to successfully complete Objective 2, and we can now move into Objective 3. In Objective 3, we're going to discuss the adaptive immune system. The adaptive immune system is divided into two branches: humoral immunity, which is driven by B cells and antibodies, and cell-mediated immunity, which is driven by T cells. Let's begin with humoral immunity. Humoral immunity refers to the branch of the immune system that uses antibodies, also called immunoglobulins, to target pathogens in body fluids. B cells are at the center of this process. B cells are going to mature in the bone marrow, and when they first encounter their specific antigen, they're going to be considered naive B cells because they have not yet been activated. Once activated, they can then differentiate into two main types: plasma cells or memory B cells. Plasma cells are the active antibody factories, and they produce large quantities of immunoglobulins. Memory B cells, on the other hand, they remain in the body long term, and they're going to be ready to respond quickly if the same antigen is encountered again. Now, antibodies, there are Y-shaped proteins that are made up of two heavy chains and two light chains that are held together by disulfide bonds. Each antibody has an antigen binding region that's also called the variable region. That's located at the tips of the Y shape. This region is going to be unique for each antibody, and it allows it to bind a specific antigen with high specificity. The rest of the antibody is called the constant region. It's the same within each antibody class and can interact with immune cells or activate the complement system. Antibodies, they can neutralize pathogens directly, meaning they block the parts of the pathogen that it would use to enter host cells. They can opsonize pathogens, which means marking them for destruction by phagocytes, or they can cause agglutination, which is the clumping together of pathogens to make them easier to clear. Now, some antibodies can also trigger degranulation of immune cells, which can cause the release of toxic compounds that help destroy the pathogen. B cells can undergo a process called isotype switching, in which they change the class of antibody they produce in response to cytokine signals from other immune cells. The major antibody isotypes are IgM, IgD, IgG, IgE, and IgA. While the MCAT does not require knowing the specific roles of each, it is important for us to understand that isotype switching changes the functional properties of the antibody without altering its antigen specificity. Lastly, the activation of B cells follows the principle of clonal selection, where only the B cells with receptors specific to the antigen are selected to proliferate and form any army of identical cells. The primary immune response occurs the first time an antigen is encountered, and it's going to be relatively slow because naive B cells must first be activated and expand in number. But then there's a secondary immune response. It's much faster and stronger thanks to the presence of memory B cells that were formed during the primary response. This principle underlies how things like vaccination works. By generating memory cells without causing disease. Vaccines essentially prepare the immune system for rapid and effective protection upon real exposure. Now, let's turn to the other branch of the adaptive immune system, which is cell-mediated immunity. This branch is driven by T cells, which mature in the thymus. And T cells play critical roles in directly killing infected cells, in coordinating immune responses, and in maintaining immune balance. T cell development in the thymus, it actually includes two important quality control steps. Those are positive selection and negative selection. Positive selection is going to ensure that only T cells that are capable of recognizing antigens presented on MHC molecules survive. Negative selection eliminates T cells that react too strongly to self-antigens. T cells that are able to pass these tests are then released into circulation as mature but naive cells that are ready to respond when they encounter their specific antigen. There are several key types of T cells, in fact. Helper T cells, also called CD4 plus T cells or TH cells, they are going to recognize antigens presented on MHC class II molecules, typically from extracellular pathogens. Once they're activated, helper T cells are going to secrete signaling molecules which recruit and activate other immune cells. Those include B cells, cytotoxic T cells, and macrophages. And this makes helper T cells central coordinators of the immune response. Unfortunately, CD4 plus T cells are the primary target of HIV, the virus that causes AIDS. And as HIV destroys these cells over time, the immune system becomes less able to fight infections, and that really leaves the body vulnerable to other diseases. Now, the other type of T cell we want to talk about is cytotoxic T cells. Those are also called CD8 plus T cells or TC cells, or sometimes CTLSs. They specialize in killing cells that are infected with viruses or intracellular bacteria, as well as cancer cells. They are going to recognize antigens presented on MHC class I molecules, which again are found on all nucleated cells. When a cytotoxic T cell identifies an abnormal antigen display, it releases peripherin, which creates pores in the target cell membrane, and granzymes, which trigger apoptosis or programmed cell death. Next, we have regulatory T cells, also known as suppressor T cells. Those play an equally important role in maintaining immune balance. So what they do is they help enforce self-tolerance by preventing overactivation of the immune system and trying to reduce the risk of autoimmune diseases. Now, without these regulatory signals, the immune system could damage the body's own tissues. Finally, we have memory T cells. These are long-lived cells that remain in the body after an infection has been cleared. If that same pathogen is encountered again, memory T cells are going to be able to respond rapidly and with greater strength, and that's hopefully going to provide faster and more effective protection against said pathogen. So together, these different T cell subsets allow the cell-mediated immune system to detect and then eliminate threats while hopefully minimizing harm to the body's own cells. All adaptive immune cells, they start as lymphocyte precursors in the bone marrow, and they branch into two main lineages, T lymphocytes and B lymphocytes. With that being said, this figure is going to help us summarize everything we've discussed thus far in a very condensed and effective way. So T lymphocytes, they leave the bone marrow to mature in the thymus, where positive selection ensures that they can recognize MHC molecules. And negative selection removes any that react strongly to self-antigens. Mature T cells can differentiate into helper T cells, cytotoxic T cells, regulatory T cells, or memory T cells. Helper T cells, they can recognize MHC class II antigens and then release signals to activate B cells, cytotoxic T cells, and macrophages. Cytotoxic T cells, they can recognize MHC class I antigens and they can kill infected or cancerous cells. Regulatory T cells are going to maintain self-tolerance and they will prevent overactivation. Finally, memory T cells are going to persist long-term for faster secondary responses. So these subtypes form cell-mediated immunity, where immune cells themselves are the effectors. Then we have B lymphocytes. They mature in the bone marrow, and when they're activated, they can differentiate into plasma cells or memory B cells. Plasma cells are going to produce antibodies that neutralize, opsonize, agglutinate pathogens, or trigger degranulation and complement activation. In addition, we talked here about isotype switching. This changes the antibody class in response to cytokines without altering specificity. This is in comparison to memory B cells, which are going to remain long-term for rapid secondary immune responses, and this is the basis of vaccination. So in short, T cells kill infected cells and coordinate immunity, while B cells produce antibodies to clear extracellular pathogens, and both of them create memory for lasting protection. Now that we've discussed the two branches of the adaptive immune system, let's look at how they are actually activated in response to different types of pathogens. The key is that the innate and adaptive immune systems, they don't work in isolation. They constantly interact. So the type of pathogen and its location in the body, that's going to determine how the adaptive immune system is engaged. So let's actually start with bacterial infections, which typically involve extracellular pathogens. When bacteria enter the body, macrophages and other antigen-presenting cells are going to engulf them through phagocytosis. The bacterial proteins are then processed and presented on MHC class II molecules. This is going to activate CD4 plus helper T cells, which release signals to recruit additional immune cells. Those signals are cytokines, and cytokines attract neutrophils and more macrophages to the site, and mast cells release histamine to increase blood vessel permeability. That is because it will allow immune cells to enter infected tissues more easily. Meanwhile, dendritic cells carry bacterial antigens to the lymph nodes where they activate B cells. These B cells differentiate into plasma cells, which produce antibodies that can opsonize the bacteria, neutralize toxins, or trigger the complement activation for direct bacterial lysis. Memory B cells are also formed. Those are going to provide us with long-term protection. Now we're going to compare that to viral infections, which involve intracellular pathogens. When a cell is infected with a virus, fragments of viral proteins are presented on MHC class I molecules, and this signals CD8 plus cytotoxic T cells to bind to the infected cell and release perforin, I'm sorry if I'm mispronouncing that, and granzymes, which induce apoptosis. Helper T cells, they also play a role here. CD4 plus cells release signals that enhance the activity of cytotoxic T cells and macrophages. Now, some viruses, they think they're smart. They attempt to evade detection by reducing MHC class I expression. But actually, that makes them a target for natural killer cells, which can detect and destroy cells that lack MHC class I. In addition, memory T cells are formed during this process so that they can provide faster responses to the same virus in the future. And this whole process of immune activation, it relies on a fundamental ability, that being that the immune system must distinguish between self and non-self. Self-antigens are molecular markers on the body's own cells that signal do not attack. Normally, immune cells that strongly react to self-antigens, they're typically eliminated during development, and that's a process that promotes self-tolerance. If the system fails, then the immune system can or may attack the body's own tissues, and that results in autoimmune diseases. On the other hand, an exaggerated response to harmless foreign substances like pollen or even certain foods, that is called a hypersensitivity reaction. That includes things like allergic responses. But autoimmunity and hypersensitivity, those reflect breakdowns in proper immune regulation. And drugs such as glucocorticoids, they can be used to suppress excessive immune activity in autoimmune conditions. Finally, I want to discuss immunization, which is the process of acquiring protection against a specific pathogen. So in active immunity, the immune system is exposed to an antigen and it mounts its own response. It generates antibodies and memory cells. And this can occur naturally after an infection or artificially through vaccination. Active immunity is long-lasting, but it does take time to develop. Now, passive immunity, that involves the direct transfer of antibodies to an individual, such as maternal antibodies that cross the placenta or antibodies given in an injection. Passive immunity, it provides immediate protection, but it is temporary because the body does not produce its own memory cells in this case. So please be able to distinguish between active versus passive immunity. And with that, we have completed our third objective, and now we can move into our final objective on the lymphatic system. The lymphatic system is a specialized circulatory network that runs alongside the cardiovascular system. But instead of forming a closed loop like blood vessels, it's made up of one-way vessels that carry a fluid called lymph. Along these vessels are small checkpoints called lymph nodes, where immune cells can monitor for any signs of infection. Now, lymph is a clear fluid that begins as an interstitial fluid, the fluid that surrounds and bathes our cells. As blood plasma leaves capillaries to deliver nutrients and remove waste, some of that fluid needs to be returned to the bloodstream. The lymphatic vessels, they collect it and they transport it through larger and larger vessels until it eventually drains back into the cardiovascular system. This return happens at the thoracic duct, which is a large lymphatic vessel in the posterior chest that empties into the left subclavian vein. The lymph nodes, they are along these vessels and they act as important filtering stations. So inside each node are immune cells such as lymphocytes and macrophages, which remove pathogens, debris, and even cancer cells from the lymph. Lymph nodes are also one of the main sites where adaptive immune responses are initiated. Antigen-presenting cells like dendritic cells and macrophages, they can bring antigens here and activate T cells and B cells to mount a defense. From here, it's important to note that the lymphatic system, it serves three major functions. First, it helps equalize fluid distribution throughout the body, and that prevents the buildup of excessive fluids in tissues, which we call edema. Second, it plays an important role in transporting certain biomolecules. So dietary fats and fat-soluble vitamins absorbed in the small intestine, they enter specialized lymphatic vessels called lacteals, where they are packaged into structures called chylomicrons and carried through the lymphatic system before entering the bloodstream. Finally, the lymphatic system is a critical part of our immune defense. It provides a route for immune surveillance. It provides a place for immune cells to communicate and coordinate responses. And It provides a setting where pathogens can be detected and destroyed before they have a chance to spread further. So, the lymphatic system is not just a drainage network for excess fluid, but it works hand in hand with both the circulatory and immune systems to maintain fluid balance, move key nutrients, and obviously protect the body against disease. And with that, we've completed this chapter. I hope it helps. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.